It is my pleasure to introduce Dr. Catharine Winstanley, who's professor in the Department of Psychology at the University of British Columbia. Hi, Kath.
Hi, Steve.
Great talking to you. So we're going to start our conversation today by talking about Dr. Winstanley's paper led by Chloe Chernoff called Neurochemical Contributions to Cue-Driven Risk-Taking and Impulsivity, published in Psychopharmacology last year. Dr. Winstanley, could you tell us a little bit about the main findings of this paper?
Sure. So the main findings are that we observed an improvement in decision-making when we administered atomoxetine, and also to some extent when we administered guanfacine. So by an improvement in decision-making, the task we used is our cued rat gambling task, and I can talk a bit more about that. And we basically are looking at both risky decision-making, so the degree to which animals will choose options that may be associated with larger per trial gains, but also disproportionately large punishments. So even though they seem tempting, they're these high risk, high reward options, they actually result in the animal earning fewer sugar pellets per session than if they chose more sort of safe, boring, conservative options that are associated with smaller per trial gains, but also much smaller and less frequent losses, which we signal through timeout periods. And overall, if the animal chooses those safer options, then they can earn more sugar pellets per session. And what we typically see on this task is that quite a few animals prefer the risky options. And part of the reason is because we've paired the wins with these very salient audio-visual cues. So there's like flashing sounds and lights that happen when the animals win. And one of the things we're really trying to sort of uncover in the lab is why basically adding bells and whistles to this otherwise fairly easy for the rats gambling task increases risky choice so much. And so we gave a bunch of noradrenergic drugs, and I can talk about why we looked at the noradrenaline system. We did guanfacine, atomoxetine, propranolol, and yohimbine. And we know that atomoxetine actually impairs decision-making in the absence of those reward-paired cues. So the animals actually go towards being very risk-averse. And yet in the presence of the cues, the animals actually now get better with atomoxetine. And we did this in males and females, which is something that we're trying to do more in the lab. And Chloe Chernoff, who's the graduate student who led this project, she was actually an undergraduate when she completed this. She was really keen on doing both sexes too. And that's a really great thing to see just in the field in general. There's a real appetite driven all the way down, top down and bottom up, but from the students a lot to want to use both sexes. So yeah, and that's, so it really makes a difference whether or not the rewards are cued. And we were quite excited by this. Obviously, both atomoxetine and guanfacine are treatments that are used and approved to try and decrease the symptoms associated with ADHD. So this might indicate that these compounds could be useful in at least some individuals who are struggling with sort of gambling use disorder.
Oh, really interesting. So one thing I've noticed from going to casinos over the years is that at least back in the day, people would be smoking constantly. So obviously that's nicotine is a drug that acts on not necessarily acetylcholine, not necessarily norepinephrine as you've been studying here, but it's very clear that there's some sort of interaction there between drug effects and the effects of these salient cues.
Yeah, we've actually looked at cholinergic drugs as well. So it seems that almost every neurotransmitter system that we're investigating, that the effects of drugs are very different if you have these cues present. So the cues seem to change sort of the neurochemical regulation of decision-making and to some extent impulsivity as well, because we can get both measures out of the task. We can actually get a measure of motor impulsivity, premature responding. It's identical almost to that you get out of the 5-choice serial reaction time task. And we did that deliberately because we wanted to see if those two behaviors, high impulsivity and risky decision-making, would track each other, because both are associated with both gambling addiction and of course drug addictions. But yeah, you're right in that the casinos always seem to be many years ahead of us, looking at the things that they allow. So a lot of alcohol, Smoking is still permitted indoors in casinos. Nicotine, we do see can increase motor impulsivity, but we haven't seen any effects of nicotinic drugs on decision making. And that's actually similar to what Barry Setlow's been finding too when he's been looking at cholinergic drugs. It's the muscarinic receptor agents that seem to modulate decision making. And again, very different effects in the presence or absence of cues. So one of my other graduate students, Graham Betts, is looking at following up on some systemic drug challenges. Again, same kind of idea, just muscarinic agonist, antagonist, nicotinic agonist, antagonist. And much to our surprise, scopolamine, which impairs decision-making in the absence of cues, which might be what we'd all expect, now improves decision-making on the cued RGT. And again, that seems very unusual. Scopolamine is not normally a drug associated with improved cognitive function. And we think it might be to do with the fact that scopolamine can decrease the sort of bias towards the most salient events. And that might be actually why it's also potentially showing some therapeutic efficacy for depression. So the rest of Graham's PhD is going to be focused on trying to unpack that, but it's story for everyone who's interested in psychopharmacology. All the most selective drugs are ridiculously expensive and don't dissolve. So we're just trying to figure out how to best pursue that. But yeah, we've got some chat-free rats, and so we can start maybe looking at some cholinergic interneurons and some selective manipulations of cholinergic pathways. But yeah, getting sort of back to noradrenaline, which is, yeah, I think, so we've seen different effects of serotonergic, dopaminergic, cholinergic, and noradrenergic drugs in the presence of cues. And we don't think it's all working through the same cognitive mechanism, but it seems that by cueing the WINs, we're sort of changing or recruiting different cognitive processes. So in terms of the dopaminergic drugs, we think this is probably related to reward learning, which is what you'd expect. But in terms of the noradrenergic drugs, I mean, there has been some really cool papers recently showing that noradrenergic locus coeruleus neurons may be carrying, like, amongst other things, a reward prediction error type signal. But typically speaking, you don't tend to get sort of effects on, perception of reward following noradrenergic manipulations. And we're wondering if instead what we're doing is changing the salience of the cues in the environment. Because we know that locus coeruleus neurons, they will fire to interesting things happening in the world. So when there's a cue, you'd expect there, like, for the locus coeruleus neurons to fire and trigger that. Like that, I mean, this is all Gary Aston Jones's stuff, suggesting that the tone of your noradrenergic system is going to affect whether you explore or exploit. And we're wondering if, when the animals are learning the task in the presence of the cues, because these cues are going to encourage phasic firing of noradrenergic neurons, that for whatever reason, the animals default too rapidly to exploitation, because that phasic firing is associated with more exploitation than exploration. So that means they're sort of fast tracking to, I think this is the right option, and I'm going to stop exploring everything else. And we're hopefully going to do more computational modeling of this task as well to actually test those hypotheses, because yeah, I'm really lucky that we've got Claire Hales with us for a couple of years, who came from Emma Robinson's lab. And yeah, she's a whiz with all kinds of different computational techniques. So we're going to see if we can get to the bottom of that a bit more. But yeah, so when the animals are learning the task, maybe the cues are encouraging them to default to what may look superficially like the best option. It's got lots of cues associated with it, larger rewards, and they're not really thinking about building a proper model of the task, which involves integrating the probability of loss and the magnitude of those losses together. So if we then give drugs like atomoxetine in particular that are going to perhaps decrease the influence of phasic noradrenergic events, because it's a noradrenaline reuptake inhibitor, it might encourage animals to go back and explore, and that might be why we're getting an improvement in decision making on the cued version of the task, which is where animals are typically making bad decisions. And yet we see an impairment on the uncued version of the task, where they're typically making good decisions because they're maybe going back and exploring things that they should have left well alone. So that's sort of our working hypothesis at the moment.
That's so interesting. Yeah, I mean, it sounds like you guys are really teasing apart the neurochemical components of gambling-like behavior. And so, like, the effects of cues on the brain recruits certain neurotransmitter systems that you guys can then show experimentally are able to manipulate the behavior and potentially therapeutically down the road. And it sounds like probably the casinos and things know about some of this too, not necessarily the neurochemistry, but the ability of cues of a certain type to kind of hack your brain and recruit these sort of really basic motivational systems that you're talking about, decision-making systems you're talking about is it sounds like it's very much intentional. Would you agree?
I think they know which machines give them the most profit, and so they can do with thousands of participants, almost like a Mendelian genetics type of experiment, just see which ones rise to the top, move them forward, and I'm sure that someone in those institutions is more than aware of how the cues are working, because when we think about electronic gambling machines, they quite often do something which Mike Dixon has been researching quite intensely at Waterloo. They disguise losses as wins. And the way in which they do that is they play a sound or show a light pattern when you win a little amount of money, but it's actually less than the amount of money you wagered. So overall, you've actually lost, but the game reacts as if you've won, and they're very deceptive. And, we all fall for this. And in fact, we've been looking at whether rats fall for this too. It's a study by Jackie Ferland ages ago. And I think the presence of the cues, at least for human subjects, seems to be really important in driving that behavior. And I think there's more and more cue options that can be added to games now. So you can do sort of more sensory immersion, you can use virtual reality, you can deliver tactile stimulation too. You can try and rather than just standing in a machine, you're almost like in a bubble. And I'm sure what people would argue is, well, that makes the game more fun and more immersive. It's like, yes, but it's also going to increase the chances you're going to lose more money. That's, our overall sort of concern. So it's an interesting, just fundamental question, just a basic science question of how does this happen? Why is it when we do something so simple as just play a little tune and flash some lights, does this really change the way our brain engages with this sort of probabilistic decision-making task? But then from a sort of public health outcome, concern, I think knowledge always helps. So if you're aware that these things, if you're gambling, that these stimuli, they aren't just there to draw your attention, they're not just there for a bit of fun, they could really be there to encourage you to lose more money, then that may change people's attitudes towards playing some of these games and how they feel about it.
Yeah, because it really kind of almost overrides your sort of deliberate decision-making strategies, which is what you guys would call good performance on the task. Like the animals should be getting as many rewards as possible, which is perhaps investing in things that are long-term rewards in a case of a human, as opposed to going to the casino and, getting some immediate wins that are with all these bells and whistles and things that kind of tap into these basic survival circuits and get you to it really taps into that basic neuroscience, I think, ultimately of how your brain is working, and it's responding in a certain way to things that are shown, your brain thinks is really important and gets you to kind of do compulsive behaviors. And I wonder if you could speak a little bit to the, people have argued that drugs do something similar. They sort of pharmacologically hack into dopamine and whatever else in the brain to kind of have this extra normal reward or something. And it's in a way, it's like hacking just like a casino would with psychology, basically, or exploiting your innate responses to cues and whatnot that are associated with rewards. Perhaps that can be done pharmacologically as well. I'm curious if you could speak a little bit to the overlap or lack thereof of drug and other sorts of addiction.
Yeah, so it's definitely true that there are hardly any individuals with gambling problems that just have gambling problems and no other either sort of axis 1 or axis 2 disorders. Depression, anxiety is very common, but so is comorbid substance use, whether that be alcohol or nicotine or more illicit drugs. So one thing that I think we as a research field need to do more with our animal models is try and combine all these things. And there was a graduate student in the lab for a few years, Tristan Hines, who followed on from Jackie Ferland's work, and they were looking at combining cocaine self-administration with risky decision-making in the gambling tasks. And one thing, Jackie really started this, It's a tough experimental design to do because you train the animals to do the rat gambling task, and then you implant them with jugular catheters and then train them to self-administer, and they get to sort of go take cocaine during the day or during the evening, and then they come back and they gamble the next day. So we're trying to look at the changes in decision making that accompany sort of early onset drug use, if you like. And she was hypothesizing, well, how does it, right from the get-go in the task, even without the cues, we have a lot of individual differences. So in the absence of cues, 85% of the animals are going to choose the best option. So even though it looks quite complicated on the surface, because we've got 4 different options associated with different numbers of sugar pellets, different probabilities of timeout periods and wins, and all that kind of thing, the rats figure it out. They figure it out within a few sessions, and then their preferences just grow over time. And yet we had this 15% that, despite equivalent training, equivalent exposure to the contingencies, they just chose risky. And so we wondered if that would change either how much cocaine they would take or the impact of taking cocaine on decision making. Because again, we know that gambling use and drug addiction, gambling use disorder and drug addiction sort of go hand in hand, at least for a subgroup of people. So it was really interesting what you found. Basically, all the animals took roughly the same amount of cocaine. At this point, we've only done, basic FR1 schedules, and that's something that Kelly Herelia in the lab is playing around with, looking at intermittent access and voluntary abstinence, all the really cool new models that other people in the field have been working on. But during the sort of sessions after the cocaine taking, so they'd all go and have some fun, take some cocaine in the evening, the optimal decision makers were just fine. They'd go back into the gambling task, make good choices, everything's fine. The risky decision makers started making more risky decisions, and more and more and more. With each passing day they took cocaine, their decision-making score, which is like a measure of overall how risky they are, just tanked. And we've repeated this a few times. So this is something that seems quite robust. And we then did some incubation of craving work. So where we just test how willing animals are to press on that drug paired lever in extinction. So we just had the cue light come on, but no drug delivered. And we compared that 24 hours after the last self-administration session and then 30 days. And what Yavin Shaham's group and others have shown is you get this incubation of drug seeking with time since last use. So the longer you're abstinent, your craving doesn't actually go away, it increases, which, as someone who is in a constant battle with nicotine dependency, is a thing, at least for me. So in the optimal decision makers, we did see some incubation of craving, but it's almost completely overshadowed in the paper by this massive increase in the risk preferring animals. And the degree to which craving incubates correlates with the degree to which decision-making score tanks during a cocaine use. So it's as if, this risky decision-making exacerbation during drug taking is associated with the chances of relapse. And that fits with some clinical data showing that, individuals who are bad at the Iowa gambling task, which is the human paradigm that our rat task was based on, that predicts treatment failure more effectively than any other measure of decision-making or impulsivity. So if you're doing badly and you don't get a recovery of that ability to sort of figure out what's a good option and to resist risky choices, the chances of staying clean really, really tank. And then we've done this again with the cued RGT, and we get a similar pattern of behavior. I mean, for some of our animals, even the optimal animals start making risky choices when taking cocaine, but certainly the risk preferring ones just get like enormously risky. They almost can't get any riskier. And we have, per cohort, we have far more risk preferring animals in the cued RGT. So if we think about that as a human population, if they're playing these very highly cued gambling tasks or games, I guess what you'd call a gambling task for human, and that's interfering with your brain chemistry, it could be leading you into, if you do then experiment with, with drugs, you could end up having a more adverse outcome as a result.
We also so interesting. Go, please go ahead.
Like get a scientist talking about the research, you can't shut them up. we've been looking at the sort of interplay as well when we start manipulating the dopamine system. So we can, if we dampen dopamine signaling chemogenetically from the VTA actually throughout the brain, when the animals are learning the task, at least in males, we can make decision making better, but then the animals go on to take more cocaine. So we're wondering if there's like an optimal dopamine balance or a stimulation balance that the animals are looking for, where, you know, you can think, oh, I'm going to dampen down gambling behavior and that's going to be good for the animals. But then if it ends up that they end up then craving more cocaine if they've been gambling, then that's, yeah, we're trying to sort of figure out to what degree these things substitute for each other or even cross-sensitize with each other. And that's, yeah, that's a bit of an open question.
Absolutely. Yeah, this is, it really brings to mind this idea of heterogeneity here in terms of just in general, like you have variation in these populations in risk-taking, a certain type of kind of impulsivity, let's say. And I think a lot of people would, at least I sort of have usually thought of the relationship of impulsivity to addiction in part due to the idea of like, do I decide to break the rules and use a drug too much or at all if it's an illegal drug? Obviously that's then going to be correlated with later problems with addiction. But your data, I think, is really showing that at least in that population that's like, you know, kind of risky in the first place, like the effects of the drugs could actually be much stronger, and it could kind of make them addicted faster, so to speak, even with similar amounts of drug use perhaps. But then also you mentioned that there's like multiple things going on here. So how much drug you take is affected by this brain manipulation, but maybe not the risky choices you make about drugs or something like that. And so, you know, it kind of brings to mind the idea that people are addicted in lots of different ways. And, you know, we really need to know those details, which is, you know, something Psychopharmacology, the journal, I think, really dives into quite a bit with, you know, in terms of your work and many other people's as well.
Yeah, I think we have to be careful not to sort of over promise on what our results mean. And yeah, I agree. There's lots of different reasons why people use drugs, use illicit drugs, and end up in patterns of problematic use, but also different reasons why people gamble. So some people gamble for the thrill and they enjoy the rush of, you know, the potential to win and avoiding losses. Whereas another whole group of individuals gamble just to escape. And they're actually, you know, generally those struggling with depression and anxiety issues. And they use these, particularly these electronic games, as just a way to dissociate. And so there's no real thrill seeking there. It's more people enter this trance-like state that Mike Dixon would call sort of dark flow, where you're just totally engaged and immersed in a cognitive challenge. But that cognitive challenge is becoming addictive and actually just, you know, taking a bunch of money out of your bank account. But for most individuals, that's the only time they've ever felt that state of flow. So it's, yeah, those populations are going to be very different in terms of their arousal profiles. And this is also something that we're very interested in looking at from the perspective of noradrenaline in particular, is that trying to understand how the cues are changing arousal, because that's clearly something that, you know, individuals who enjoy gambling are reporting that they, you know, they get changes in arousal. And there's a lot of human research trying to measure those changes and look at things like heart rate variability and pupil diameter changes. Pupillometry is a bit of a challenge to do in rats, but we have started working with some telemetry implants. And yeah, this is more work actually by Chloe and Claire. And we're seeing that in the queued version of the task, even with a really small number, because unfortunately this research is horrendously expensive to do. But I think even with really tiny n's, you can see the queued animals are just very different. Yeah, they get a lot warmer, their body temperature goes up, even though they're not moving around that much more. Their heart rate variability is very different. It seems this is why we need a grant to actually get at this, because I didn't realize how sensitive rats' heart rates would be to moment changes on task. But in the unqueued task, it seems there's lots of things happening, like the heart rate spiking and going all over the place. But we can't, with the equipment we have, because we kind of like found some of it in the bottom of a drawer kind of thing, and we're borrowing bits and pieces, we can't sync the Metassociates code to the heart rate data yet. And now I really want to do that because it looks as if whatever's happening on task is really influencing the heart rate of the animals in the uncued version in a way that it doesn't seem to in the cued version. But that's all a bit speculative. We haven't definitively showed that yet. But yeah,
I really like that idea, and I understand. I mean, I sort of I understand that humans also respond to cues with heart rate increases as well, at least for smoking related cues and things like that. So I love this idea of another data stream coming in. If because this could not only tell you something about like what do cues do to psychology, behavior, the brain, the body, but also perhaps like maybe some of the animals don't have a heart response to cues and maybe they're different,
Right,
and perhaps
exactly.
atomoxetine will affect them differently and so on.
Yeah, and that is something that I should, even there are animals on the cued task that despite the fact that generally population level wise, we see increases in risky choice, there are animals that perform just as well as the animals on the uncued task. It's not that every single rat goes risky. And so one of the things we are really interested in is looking at those guys too. Like, how come these guys are so good? They seem their ability to resist all our temptation signals and just carry on making really good decisions. Something that Kelly Horelia has been doing too is looking at preference between the two tasks, which is tough to train. But again, they learn that left lever is uncued task, right lever is cued task, and look at individual preferences. And then look at the decision making on their preferred or unpreferred option. And that's turning into a really exciting story. And just sort of to summarize, like it seems like a lot of the cued rats, the animals that prefer the cued task are also the animals that make risky decisions. And the more risky decisions they make, the more they prefer the cued task. And whereas the guys that prefer the uncued task, they're making slightly better decisions and they're also generally less impulsive. So there's, just factoring, it's a huge data set. Again, we're going to be really relying on computational modeling to help us interpret that in the most optimal way, but I'm really excited about it. I think it's gonna be really cool.
I think that's a great plan. And I can just say from my own personal experience, as I've mentioned, I've been to casinos before and I do not really like gambling. And I suspect that's because in part, the first several times that I've been to a casino, I lost what I had, which was no money at the time. I think I was a graduate student or something. And I didn't like that at all. And I think I'm very responsive to that punishment, in other words. And I wonder if your rats, if screened for like sort of the negative aspects, like how sensitive to chance, like, how sensitive to like probability or perhaps even to some kind of a punisher. I wonder if you might also reveal other types of individual differences.
Yeah, that's a really good idea. That would be a really cool thing to do. We're also hoping to get fiber photometry working so we can see what their sort of dopamine or noradrenaline responses are really early on in the task. So, are the animals that are gambling, do they have, or they like the risky options, do they have a blunted dopamine response to small wins, or is it like a hyper dopamine response to large wins? Like that's going to be kind of interesting to figure out. And just figuring out that individual difference piece, I think is going to be so important. And I will say that it's not just rats that show this cue-induced behavior. Human subjects show it too. So we've used a sort of two-choice lottery task in our lab to show that, people prefer making risky choices when the wins are accompanied by sound and light cues. And also other folks have shown it. Elliot Ludwig has shown it when she was working with Marcia Spatch using a different kind of task. So it's, sadly, we're all, not necessarily better than the average rat when it comes to some of these things.
Yeah, and that's probably something that the sort of gambling industrial complex has known for, a lot of time. Similar in some ways to, we knew there was a lot of things known about nicotine and its effects on Jews and whatever else by the tobacco industry that we are now rediscovering, I think, these days. So this is this has been a really very interesting conversation, Dr. Winstanley. A lot of implications for human behavior, which I love, and I think your work really kind of exemplifies the spirit of the journal in many ways, like carefully deconstructing behavior and the brain mechanisms or neurochemical mechanisms, at least thereof. So I appreciate you taking the time and for discussing this this paper from 2021, Chernoff et al. And thank you again for your time, Dr. Winstanley.
Thanks very much. It's been an absolute pleasure.